Hi guys, so in the last class we started with rDNA technology. Now we are going to complete the part, okay. So the rDNA technology, the last part of the chapter, the last part, so few points are left that we are going to discuss, right. So rDNA technology, recombinant DNA technology, or we call it as cut and paste technology, genetic engineering, right. So in this we have already seen some points. What were the points? First was isolation of DNA. So from the cell wall everything is removed and the DNA is isolated. So that was first point. After isolating the DNA, restriction enzyme was added. The DNA was made into many fragments. That fragments were put into which one? Electrophoresis. Then that particular gene of interest was taken by process called as elution, isn't it? So these are covered. Fragmentation is done. Isolation of the desired gene, right? So that is taken. Gene of interest is taken. And now this gene of interest was directly not put into a vector. Before that we are going for amplification, where we are going to amplify. Amplify means what? Make many number of copies of that particular gene that was carried out. So that one we call it as what? Amplification. So amplification of the gene of interest was done with the help of a process called as PCR, polymerase chain reaction. One gene of interest which I am getting, from that I am making many number of copies of the same gene. So that process we call it as what? Amplification. Why? So that I can make many number of rDNAs, as well as if one is lost, no harm, because I am having billions with me. Now after amplification, what is the next step? Ligation, isn't it? So the plasmid is collected, same restriction enzyme is added, cut is made. In that cut, what we are introducing, in that cloning site I am going to introduce my foreign DNA and ligating it. Ligating, you know, with the help of DNA ligase, joining it. Then after joining this one, what we are going to do? We are going to put it into the vector, isn't it? So either in the vector or we are going to put it into a bacteria, or we will be putting in plants where it has to take its function. So transferring that into the host. For transferring that into the host, we discussed some of the points. So what were the points? The first point was by chemical means. By using calcium ions, heat shock treatment and all, we can allow them to enter into the host, that rDNA into the host. Otherwise, we can use micro-injection, ballistic gun for introducing them. That is somewhat physical. Or we can use disarmed pathogens, pathogens which are causing disease in that organism. The pathogens are introduced by taking out the disease causing factor and introducing my gene of interest I am introducing into it. So that in competent host we discussed physical, chemical as well as the biological method, so that the rDNA is entering into the host. So that is transfer of rDNA to the host. So amplification PCR we discussed, vector we are ligating to a vector, rDNA is formed, that rDNA is transferred to the host. Listen students, today we are going to discuss about the last part of the topic, rDNA technology, culturing the host cell on a large scale, extraction of the desired products and downstream processing one by one. Now I will tell you what is actually we are going to do, culturing of the host cell on a large scale. Now this rDNA, if it is an insulin producing gene of interest, is put into that plasmid, which is that the vector, what is present? The insulin producing gene is present. Imagine, okay, any enzyme producing, insulin producing, hormone producing gene is present. Now this rDNA, where I am going to introduce is that I am going to introduce into the bacteria. So the plasmid from the bacteria is taken out. In that I have made a cut. There I have introduced my foreign DNA, that is insulin coding gene. Now what is happening? This gene, actually this is acting as what? It is acting as an rDNA. This rDNA, where I am going to put it? I am going to put in this bacteria, okay. So by any physical method or by bacteriophage, somehow I am putting it in, okay. So after putting it in, this plasmid has been put in, rDNA is put into in. Then what I am going to do is that I am going to culture this bacterial cell. Culture means what? I am going to allow this bacterial cell to multiply, right? So now what is happening? That rDNA is put there. So in this bacteria the rDNA is put. The bacteria is dividing as well as the plasmid is dividing within itself. So now the insulin producing gene is also dividing within itself, isn't it? So like that, the insulin producing gene is dividing within itself, and all the bacteria are coding for what? Production of insulin. So they will be producing what? Insulin. So these host cells, this is the bacterial host cells, are cultured so that they are producing what? Lot amount of insulin. Okay, why they are producing insulin? Because in that bacteria I have introduced the gene which is responsible for coding for production of insulin. So insulin will be produced. So in this medium you can find much amount of insulin will be present. Now what will be the next step? So this is culturing. First step was what? Culturing. I am culturing this bacteria, making to multiply and grow in number. Thereafter they are producing insulin. That insulin is taken out. That process from this medium, what is taken out? Insulin is taken out. So this process we call it as what? Extraction of the desired product. After that, what we are going to do? Downstream processing. Immediately after extraction of insulin, may not be it is in pure form. It has to be given to patients who are suffering from diabetes, isn't it? So I have to check whether the insulin what I have got is in good quality or not, if it is contaminated or not, if it is okay or not. So in downstream processing, after extracting this insulin, I will be adding some preservative. Thereafter I will undergo some clinical test to confirm the product what I am obtained is correct. Then I will release it as a medicine. I will pack it properly in a bottle, seal it, okay, and thereafter name it. All labeling and all will be done. It will be made germ free. Then as a packed product, it will be released out. The whole steps we call it as what? Downstream processing. What do we call them? Downstream processing. So what I have got? I have got insulin. How I have got this insulin? By this rDNA technology. Okay. So I am making insulin with the help of these bacteria. So how I am making it? I have introduced rDNA into the bacteria. The bacteria is going on dividing. Culturing is taking place. When they are dividing, insulin producing gene is helping in the production of insulin. Insulin is formed in the medium. That insulin is taken out, okay, extracted, and that insulin is treated properly, checked properly, preservatives are added, and as a final product it is given out from the factories. That process we call it as what? Downstream processing. For doing all these three steps, there are some machines available. That one we call it as bioreactors. What do we call them? Bioreactor. So this is an example of bioreactor. So the rDNA which I have produced, where I am going to put it is that I am going to put in this rDNA. I have put into this bioreactor. In this bioreactor, what is happening? Here the nutritive medium is added for the growth of the bacteria. The rDNA containing bacteria, all nutritive medium is properly added. Thereafter, here sterile environment means germ free, no other organism can enter. That is also taken care. Acid base regulation is being done. How much acidic should be a basic medium, what is proper for them, like that it is maintained. Temperature is properly maintained. Pressure is properly maintained. Foam formation, because in this reactor actually what is going to take place is that some reaction is going to take place. Okay, they are going to rotate, floating type. So some reactions, air is being pumped, agitated. Agitated means they will be moving to some extent. So when all these processes are taking place, sometimes foam will be formed. Foam, isn't it? So when you apply soap, foam comes out, isn't it? So like that, foam and all will be formed. So it should not allow the formation of foam. For that, foam control is present. Okay. So like that, everything is maintained in this bioreactor. There are two types. This one is simple stirred bioreactor, and this one is packed stirred bioreactor. Here what is the advantage is that here gaseous supply, okay, gas, when you are saying sterile air bubbles are pumped in. So the oxygen supply will be more compared to this. Here agitation is there to some extent, oxygen is there, but here the oxygen supply is more. So when this bacteria is here and all the necessary conditions are provided, what will happen to this bacteria? They will go on multiplying. So when they are going on multiplying, the gene which is present in it will be coding for what? Many factors. Along with that, they also will be coding for what? Insulin production. So the insulin is being produced in this medium, isn't it? So at a time, what is happening is that I am taking out small plots. I will be taking it out and I will checking whether the insulin is produced properly or not. Okay, so after seeing that it is not contaminated, checking and all is done, insulin is extracted out from this what? Bioreactor. Like that, what I am getting the insulin. So this process we call it as what? Culturing and extraction. After that, we will be going forward downstream processing. Now we will just see what they are saying about this process. Okay, obtaining the foreign gene product. The foreign gene get expressed under appropriate condition. The cells harboring cloned genes, so that is rDNA, can be grown on a small scale in the laboratory. Okay, so in the small scale they are growing in the laboratory. Thereafter, that bacteria which are grown in the laboratory are in the bioreactor, they are called extracted desired protein. What is that protein there? Insulin. Okay, and they are purified. I told you, adding preservatives, doing all sort of tests and all, then they are releasing it. Okay, next is that the cell can be multiplied in a continuous culture system. So in the bioreactor there is a continuous culture system wherein the used medium is drained out. So medium, when we are applying, when you are saying that some amount of food is taken by the bacteria for multiplication, food is lost, they are taking out the used undigested, every part is being removed and the new food is again added. Periodically they are changing the culture substances, the food is being provided. Like agarose gel they will be growing, means the agarose gel, sodium, potassium, calcium, protein, everything is being provided so that the bacteria grows very nicely. Fresh medium is supplied in between. Okay, is added from the other to maintain cells in physiologically most active form. So the bacteria are physiologically very active and they go on multiplying, and there is an exponential phase. Means what? You know exponential curve, isn't it? They grow on fastly because every food, everything, resources are fully given there. So there is no shortage of resource. All food is provided to the bacteria in the reactors so that the bacteria goes on multiplying. So in the bioreactors you can find this much liters of culture can be produced. This much amount of process can be done, right? So bioreactors can be thought of as vessel. Raw materials are biologically converted into products. Okay, like enzymes, using what? Using microbes. A bioreactor provides all optimum condition for producing this product. So what is the product? The product is the protein, insulin, isn't it? So here there is temperature, pH, substrate, salt, vitamin, oxygen, everything is maintained at a proper thing, and it is mostly stirring type. Stirring, I told you, they keep on moving so that air is being supplied and they are agitated. Stir tank container is usually cylindrical and they are having what? A curved base. So base is much curved. Okay, so that mixing will be proper. If the base is much curved, then the mixing will be proper. If it is flat, the mixing will not be proper. So in order to mix it, mix this one, the culture medium, actually the base is what? It is curved. Okay, the stirrer facilitate even mixing of oxygen availability throughout the reactor. Okay, the bioreactor has what? Oxygen delivery system, foam control system, temperature control system, pH control system. Sampling pots is what? In between we can take out some sample and check whether there is proper working taking place, whether there is any contamination with any microbes or not. For that also facilities are available. We can take out small pots and then we will check it out whether my process is taking place properly or not. After this culture is extracted, now with many different physical processes, many in the culture other substances will also be present along with the insulin, isn't it? But I want what? Only the insulin. So in order to go for that, I will be going for the last process called as what? Downstream processing. In downstream processing, what is happening? Filtration, purification. Okay, all things are filtered correctly. The insulin is being taken out, purified out through various processes which we are not going to discuss. Okay, so the insulin is taken out and they are added with what? Suitable preservatives. And after adding suitable preservatives, many tests is done whether the insulin is fit to be marketed or not. Like that, many clinical tests is done. If it is a drug and all, you have to definitely do what? If I am producing drug from some rDNA technology, means drugs and all should definitely go through what? Clinical test. After the test, if it is confirming that it is safe to use, then all quality control measures and all is being given, batch date, thereafter manufacture date, everything is given, and it is released as what? A new product. It has been released into the industries. So this one we call it as what? Downstream processing. Final marketable product like that to release, what all steps I have to undergo after the extraction, we call it as what? Downstream processing. Okay, I have got the thing, now I have to correctly purify that thing and make it very safe and then release it as a new thing. So this one we call it as what? Downstream processing. So a quick recap we will do. So what is a bioreactor? So 3 steps we are going to do. First is what? rDNA has been introduced into the host. Here I am telling bacteria is the host, because in this bacteria rDNA, which is coding for insulin, is present. Insulin actually it is not done like that, but I am taking so that you can understand easily. So this insulin is present, rDNA is present in that bacteria. That bacteria is introduced where? Into this bioreactor. What is bioreactor? Bioreactor is a machine in which all temperature, pressure, foam, oxygen supply, everything is controlled in a very proper way, right? And thereafter you can find here culture medium is put. What is culture medium? For the growth of bacteria, what and all nutrient is present, that in a form of a solution they are mixing it and properly introducing it into the bioreactor. What will happen when the nutrients are present? The bacteria will start to multiply, they will grow in number, and all these bacteria will have what? That recombinant DNA, and they will be producing what? They will be producing my product. What is the product? My desired product is insulin. They will be producing. Okay, so that production is done. So in order to, when they are producing this insulin, in between what I will do, sampling pots are there, I will be checking whether correct amount of product is formed or not, whether it is contaminated with any other microbes or not. Everything is being under checked. Okay, in between frequently they are checked. Afterwards, when the nutrient medium is becoming less, again the older medium is taken out and the new culture medium is again introduced into the bioreactor, so that whenever food is needed, food is supplied. So you can find the bacteria is growing in very high speed. Exponential phase of growth is taking place. All these bacteria will produce what? They will be producing insulin. Now in this medium, insulin is present along with other substances. The last one what we have to go to is that I have to go for downstream processing. In that, what is done? Out of these product what I am getting, I am separating, filtering and purifying only the insulin out. After taking out that insulin, I am adding some preservative, I am undergoing some test with this product, and thereafter I am sealing it in a container and releasing it as a medicine or as a product into the industry. Okay, so this process we call it as what? Downstream processing. So rDNA is helping in the production of insulin by this method. Hope it is clear. Thank you.